Hi guys, this is Dr. TS, and today I'm going to be finishing out your lectures on the scientific method. So far you've heard from Professor Pfannenstiel all about the scientific method so far. So you've talked about observations, hypothesis predictions, and experimental design. And so today we're going to finish that out by talking about what do you do with the data that you collect and how do you make conclusions based off of that. So when you're doing an experiment, you are collecting data in some way, shape, or form. And we can define data as any raw collection of unorganized facts, numbers, observations, or symbols. And there are two major types of data that we can collect. The first is quantitative. So these are numerical values that can be mathematically calculated or measured. So this could be distances, times, or sales figures. So one example of this is if we wanted to compare the height, right, of two people, of multiple people. We could measure how tall they are, and we'll get numbers, right? 64 centimeters, 20 centimeters, 140 centimeters, right? We get numbers that we can then compare. We also have qualitative data, and these are going to be categorical information that describes a trait or categories that can't be easily measured with numbers. So an example of this, maybe you want to do a study on how happy are college students, right? That's not something that you can quantify with a number, right? So we call that qualitative data. We can still collect it and we can still analyze it, but they're just two different types and they both kind of have their own positives and negatives. So once we collect the data, we then have to turn it into something that is usable. So why do we analyze data? Well, first, let's define data analysis. Data analysis is defined as the process of reviewing and exploring data to help us draw conclusions and answer our research questions. And we generally do this using math, right? We don't just kind of do it based on our interpretation. We usually use math and other processes to help us. But typically, we just get a bunch of data and we then have to analyze it. So in this example, what if I asked the question, what fertilizer makes plants grow the tallest? Okay, maybe that was my research question. So then I run my experiment, right? I have a no fertilizer category, it's my control. I try Miracle-Gro and I try Dr. Earth. And I measure these plants, I grow them for 10 days and I look at their height. And I get a bunch of different numbers, right? And it can be hard to look at these numbers and tell me which one's the best, right? Try it right now. Try to look at these numbers and tell me which one is better than the other. It's hard to, right? Because you're just looking at numbers. And so a big part of data analysis is taking these numbers and turning them into something usable that we can actually see. And so there's kind of three main goals of data analysis. The first is we want to be able to identify trends and relationships, right? Which fertilizer works best? A huge part of this is testing the hypothesis, right? If I made the hypothesis that Miracle-Gro would work better or that no fertilizer would work better, how is the data supporting or not supporting my conclusions? And finally, a huge part of data analysis is actually to reduce bias. Now, we're not going to go into too much detail and talk about the nitty-gritty of how we analyze data and how we do this to reduce bias, but if we use math and programs and such to help us with the data analysis, that can help reduce our own bias, right? I'm not just looking at these numbers and I'm like, oh, well, Dr. Earth has more numbers in the 60s, so it's better, right? That's a biased approach, right? Data analysis allows us to do these as unbiased as possible. So once we get all of this data, we then need to be able to show it in different ways, and we then put them into graphs or figures. And there's going to be three major types of graphs that we want you guys to know for this course. The first is a bar graph. Bar graphs represent the average response for all individuals within a treatment group. So if I go back to this slide, right, I pretty much took the average of each of these. So I averaged all of these together and I tried to get the average height for the plants. An average just means you add all of your numbers together and divide by the total number of values to get that number. And so the bar graph, right, so our no fertilizer was an average of like 41 centimeters, Miracle-Gro was 52, and Dr. Earth was 55. Right? Those are the average heights of each of these plants. And this is really good for demonstrating differences at one discrete point in time, right? We're just looking at a number, the average height of these plants after 10 days of growing with this fertilizer. So what is really cool about bar graphs too is that the size of the graph is directly proportional to what it's showing us, right? We see that the no fertilizer group is a shorter bar. We can immediately tell visually that no fertilizer plants grew shorter than our other plants did. However, there's one thing that we have to add to bar graphs that's really important, and that means we need to show the variability. So if I go back to this image here, right, I can't remember the averages, it is 41, 51, and 55, okay? So this is the average, but you can tell some of these numbers are higher and lower than that average, right? We call this variability. So another way to show this, and I'm going to use another example here. So let's imagine we're running another experiment and we're interested in how big these plants are getting, and we're exposing them to sun or no sun. So let's say we do this experiment, we look at 4 plants. We take 4 plants, we put them in the sun, we take another 4 plants and put them in no sun, okay? And we can calculate the average height that they grow. However, if I do this experiment again, what if I see this, right? My averages are still the same, but I have a huge amount of variability, okay? The averages are the same between these 2 experiments. So my conclusion in general would probably be the same. The sun is gonna make them grow taller. But you can tell, right, in this experiment, all my suns were about the same size, all my no suns were about the same size. However, the second time I did the experiment, some got really big, some were really small. They still averaged out to the same number, but my graphs aren't showing the variability that was present in the experiment. And so in order to show variability within an experiment, we use something called standard deviation. And whenever you see a bar graph or even a line graph, it is super important that you look for the standard deviation, because all experiments that we do have some level of variability, right? Not everyone is going to grow at the same rate. Not all people are going to respond to a medication the same way, right? Variability is a part of nature, and so we have to be able to show that. And so we use standard deviation, but it's just a math term for variation. So how far away were numbers on average from the average? It's a measure of how spread out the data is. So here, even though these 2 graphs, both of these this has an average of 10, this has an average of 25, same here, right? The variability is vastly different. So if I were to ask you right now, just looking at this experiment, which data would you maybe trust more, right? Probably the one with the least variability. But this is why it's important that you are looking for these things when you're looking at data that's being shown to you. The next type of graph is a line graph. Line graphs are used to denote changes in a dependent variable over time. So let's go back to our fertilizer example. So instead of looking at the plants at the 10 days, what if we measured their heights over the 10 days? And we could see how these changes are occurring over time. And there's a couple different things that we can look at. We can see how quickly, right? How quickly it's growing, and also the direction that it's going in. So we call this slope and direction. So remember slope, right? If it's a steep slope, I mean that change is happening faster, right? However, if it's a slower slope, the change is happening slower. They might end up at the same spot, but the change is happening at different rates. So again, this is another important thing that you can kind of see just by looking at the graph. Additionally, line graphs can tell us whether we've gained something, lost something, or had no change. So, right, we're obviously increasing in this example. We can lose something, or there can be no change over time. And all of these are telling you things about your experiment, about your data, in a way that your brain is able to interpret it in a much quicker method than if I just gave you a list of numbers and asked you, what direction is this moving? Is it going positive? Is it going negative? Is it happening quickly? Is it happening slowly? Right? The whole point of putting our data into these graphs is to make it easier to interpret and easier to really see the relationships between our variables. The last type of graph I want to talk about is something called a correlation. Now, correlations are used to determine the relationship between two quantitative variables. So this happens when you pretty much take, two variables. So in this example, we have height and weight. So let's say, for example, someone is 60 inches tall, okay, 60 inches. And we asked them what their weight was, and they told us 150. We would then put it on the graph. At 60 inches, they're 150. And you do this for a bunch of people. So if I did this with all of you in here, I had you tell me your height and your weight, I could then graph, and we could see that height and weight seem to have a relationship. And then we could kind of have it draw a line and tell us how correlated, how related are height and weight. Correlations are all about the relationship between two variables. Now there's different types of relationships that we can see. So this is all going to be based on the direction of the line. So some relationships are positive. So as one is increasing, the other one is increasing. We can have negative correlation. So as one is decreasing, the other one is decreasing. Or there can be no correlation. So maybe the independent variable is increasing and the dependent variable is decreasing. So again, looking at this just straight up, you can start to see some of these relationships and trends and make some conclusions. We can also look how strong of a correlation, so how strong is the relationship between these two variables. And we do this by calculating something called the R-squared value. Pretty much what this means is it gives us a number between 0 and 1. The closer that number is to 1, the stronger the relationship is. So we will say something has a strong correlation if it has a number closer to 1. If it has a weak correlation, it has a number that's lower, and no correlations are going to be close to 0. So this tells us how strong the relationship is between two variables. Now it's really, really important when you're thinking about correlation that you recognize that correlation does not equal causation. Just because two variables have a relationship with each other does not mean that they are causing and affecting each other. So I would like to kind of bring up two funny examples here. So here, for example, this is looking at the relationship between the associate's degrees, okay, a 2-year degree awarded in a liberal arts school like Washburn, and Google searches for tummy ache, right? Just based on this graph, you can see that these are two positively correlated relationships. The number of liberal arts degrees is positively correlated with Google searches for tummy aches. Do we think that liberal arts degrees are causing tummy aches? Maybe. You might argue that by the end of this class. Do we think tummy aches are causing liberal arts degrees? Probably not. However, there is a positive correlation relationship between the two. Another example, there is a meme, right? The meme, we live in a society, right? You've looked at how popular this meme is, and we can correlate it with wind power generated in Namibia, right? Two completely different things, but if you were to graph them and look at their correlation, you would see that they are positively correlated with each other. So just because two variables have a relationship, and that relationship might be strong, does not mean it's a cause and effect relationship. They're not causing and affecting each other. We're simply looking at the relationship. So how do we take all of this and turn it into a conclusion? And so when we look at our data, and usually we collect multiple different types of data and look at lots of different experiments, right? But the whole point of it is we want to make a conclusion. And there's kind of 3 outcomes that we can have. We can have the outcome that our data does not support a hypothesis. We can have the outcome that our data does support a hypothesis, or that the data is inconclusive. These are the 3 potential options. And all are going to have varying, and sometimes you can have one hypothesis be supported and another not. So these are all potential verdicts. What's really important, once you kind of make that conclusion on your response, you then want to share it with someone. Now, sharing conclusions is actually a really essential part of the scientific process. There are hundreds of thousands of scientists in this world, and we all work on different things. And so in order to share that information, we publish it in peer-reviewed journals, meaning you take your scientific method, you write up everything you did, you submit it to a peer, another scientist who understands the type of science that you're doing, and they make sure that it's a valid scientific experiment. They're going to ask, are you controlled? Are you unbiased? Did you analyze your data the right way? Did you make the right conclusions? They're checking your work, because if you're about to share this with people, you want to make sure that it's the correct conclusion. So peer review is a really important part of this. And then once it's accepted, it gets published in journals like Nature or Science, lots of different types of journals. Now, if you do enough of these, right, if you do enough experiments on something, then it can end up in a textbook or as part of a scientific theory. So repetition is essential in science, right? When we make a conclusion and we say, okay, X causes Y, right? We want people to repeat this. The more we show evidence for a hypothesis, the more confident we feel in it. So typically just one paper or even 2 or 3 or 4 is not enough to take it from a hypothesis to a scientific theory. So something only kind of becomes a scientific theory when it has been repeated by lots and lots of different people all over the world and is widely accepted by the scientific community, right? The scientific community has kind of come together and said, yes, we agree with this conclusion, right? The stuff in your textbook, the stuff we're teaching you, are things that have been agreed upon by the broader scientific body that says, yes, we have enough evidence that this is how this process works. And so sometimes people use theory, kind of informally, like, oh, I have a theory about this or I have a theory about that. But in this class, when I say scientific theory, I mean there's the scientific word for it. This is a theory, this is an idea that multiple scientists have studied, that there's been lots of evidence for. And so we feel good and can trust it. All right, thank you guys. Have a good rest of your day.